Thank you to the organisers for inviting me to present this work on the transcriptomic changes in ALS astrocytes. ALS has traditionally been regarded to be due to selective motor neuron pathology. However, accumulating evidence implicates astrocytes in ALS pathogenesis. I have no conflicts of interest. I searched for all publicly available RNA sequencing datasets of stem cell derived or iPSC derived astrocytes from ALS patients. I found 4 datasets with mutations in VCP, SOD1, C9orf72, and FUS. I first examined each mutation individually and found only a small number of differentially expressed genes for each mutation. But because we were interested to know what changes were common across these mutations and across ALS astrocytes, I pooled them and performed a meta-analysis comparing all ALS astrocytes with health controls. There were a substantial number of differentially expressed genes, with 1,300 upregulated, shown in red, and 1,500 downregulated, shown in blue. Gene ontology enrichment analysis showed that genes increased in expression in ALS astrocytes were enriched in ER stress and extracellular matrix, whilst genes decreased in ALS astrocytes were enriched in synaptic and neuronal terms. The glutamate uptake gene set was strongly and significantly decreased in ALS astrocytes. Because of these inflammatory properties of ALS astrocytes, we were interested to know how they compare to neurotoxic astrocytes that have been treated with TNF-alpha, interleukin-1 alpha, and C1q, also known as A1 astrocytes. I searched for all publicly available iPSC-derived astrocytes that had been treated with these pro-inflammatory cytokines and meta-analyzed them in the same way as I did for the ALS astrocytes. I then compared the ALS and A1 astrocyte signatures. Comparing the gene expression changes in ALS and A1 astrocytes, I found a positive correlation, and there was a significant number of overlapping differentially expressed genes. The 381 co-regulated genes were enriched in gene ontology processes, including ER stress and extracellular matrix, and the 497 co-downregulated genes were enriched in neuronal and synaptic terms. This supports the hypothesis that ALS astrocytes acquire neurotoxic features. This schematic of healthy astrocytes in left on the blue and ALS astrocytes in red on the right summarizes our findings. ALS astrocytes display transcriptomic changes consistent with pro-inflammatory cytokine-treated astrocytes with impaired glutamate uptake, ER stress, and immune activation. This work is now being published in Genome Research, where you can find all the detailed results.